A UTI is a urinary tract infection. This infection impacts an estimated 50% of women, and 30 to 50% of these women will suffer recurrent UTIs. A UTI occurs when the bladder is continuously invaded by bacteria, which can accumulate to substantial numbers before clearance. Antibiotics have been traditionally used to treat UTIs, but antibiotics have drawbacks as E. coli becomes antibiotic resistant. Additionally, antibiotics upset the natural bacteria needed for regular bodily functions. This experiment made use of the scientific method and strong inference we discussed in Module 1 of Biology 1510. The researchers first hypothesized M4284 could be used to treat UTIs instead of antibiotics, and then devised a test to invalidate that claim. Pili on the E. coli adhere to specific carbohydrates on receptors and begin to invade the cells of the bladder lumen. This single bacteria then divides and forms an intracellular bacterial community, or an IBC. In order to establish an effective way to combat UTIs, a strain of UTI-causing E. coli must be chosen for testing. It is important to determine that the strain used for testing is representative of all the strains so that any treatment tested on the representative strain will work for all the strains of UPEC. To determine the similarity between the strains, similarities were examined in the DNA sequences of 43 different strains of E. coli, collected from 14 women with UTIs. Bacteria with an F17-like pili were chosen for further in vivo testing. DNA sequencing uses ideas from the DNA and genetics section of Biology 1510. A similar DNA sequence means that the DNA will be transcribed to RNA, and the RNA in turn will code for proteins, which is the central dogma of molecular biology. Additionally, a similar primary sequence will lead to a similar structure and therefore a similar function, because in Module 2 we learned that protein structure dictates function. In Module 2, we learned about competitive inhibitors, which compete with the substrate by binding to the active site, so the enzyme cannot carry out its normal reaction with the inhibitor. Researchers found a competitive inhibitor, M4284, to block adhesion to host gut lining cells. They checked to make sure important probacteria were not affected significantly. Mannosides effectively reduce E. coli concentration, which decrease rate of UTIs. In an in vivo study, the competitive inhibitor was added, and the amount of colony-forming units of UTI-causing bacteria significantly decreased, while not significantly altering the beneficial microbial community when studied within mice. The impact of this experiment is wide-reaching. The competitive inhibitor protein, M4284, can be used to prevent UTIs in women, improving overall health. Additionally, it is cheap, easy to produce, and doesn't disturb the overall microbial community in the gut. Thank you for watching. Here are the sources we used to make this video.